Welcome to the podcast of Hemisphere, the official journal of the European Hematology Association. Hemisphere's podcast presents insightful, expert discussions about recent hematology publications. We hope you enjoy.
Hello and welcome to the Hemisphere podcast. I am Charles de Bock, your host today, and I'm joined by Professor Roland Koper and Dr. Jan Loofen from the Prinses Maxima Center for Pediatric Oncology in Utrecht, in the Netherlands. You are both the lead authors on a new publication in Hemisphere, which is entitled Notch1 Fusions in Pediatric T-cell Lymphoblastic Lymphoma, a high-risk subgroup with CCL17 or TARC levels as a diagnostic biomarker. So welcome to you both.
Thank you.
So I'll get straight into it. In the current paper, your focus has been on T-cell lymphoblastic lymphoma. And I guess, can we perhaps start with you, Jan, to give us a bit of a brief overview on what the clinical unmet needs are for these patients and children in terms of outcomes and prognosis?
Well, the T lymphoblastic lymphoma patients, I think in the Netherlands, it's a quite rare disease. I think in the Netherlands, we see about 10 to 15 patients, children with the disease annually. And I think about 20 ALL patients. So it's a relatively rare non-Hodgkin lymphoma subgroup. They're currently treated according to the ALL-based strategies and have been for a long time treated according to a protocol which is used in Europe. Mainly it was the Euro 02 protocol, about 20 years, something like that, with a little bit with few improvement in treatment strategies. So ALL-based treatment, 85% event and overall survival. But for the relapse patients, so I think 15-20% of our patient group, we have relatively few options, little options. So in contrast to the T-ALL patients, which we can stratify based on the MRD or some high-risk hallmarks like ABL1 fusions or things like that. But for T-LBL, we for a long time had no treatment strategies, just solely based on the stage of the disease. And we divided them in the previous protocol on stage 1-2 disease and stage 3-4 disease. You have a mediastinal mass, which is almost always the case in T lymphoblastic lymphoma. You are already stage 3, then all patients were treated quite similarly. So 85% event and overall survival, relapse rate 15% with few cure rates. That was for us a very important group to investigate. Why are they relapsing and what can we do about it? So in the last protocol, we started in Europe in 2018. It was the LBL 2018 protocol, right, based on Notch1 DNA or FBXW7 mutations. So if you have mutations in one of those genes, in specific domains, then we know, based on studies done by Birgit Burkhardt, but also by others, that it has a good prognosis. So we divided the patient group into subgroups, the ones who have mutations in the FBXW7 or in the Notch1 specific domains, DNA mutations, and they were treated according to our standard strategy. The residual group, for which we do not know what the exact drivers are or the exact mechanisms are, we put high risk group, because if you look in the complete cohort of that group, then you know that the prognosis is quite worse. So we divided them, we treated them, we randomized them according to these molecular aberrations. If you had the Notch1, you were FBWX7, you were standard, and the other were high risk, and treated either according to the standard risk arm, or we added 2 high risk blocks in the middle of treatment. And I can add just a little bit on that thought later on. But for us, it was important that we wanted to know why do these patients with unknown genetics relapse, and are they all a little bit more risky, or are just a few of them which downgrade the complete cohort to a lesser prognosis? So that was the initial drive to focus on this specific project.
Yeah, because I think, from a diagnostic perspective, when I was speaking to Roland before this, in terms of TLL and TLBL, really it's kind of a way that we categorize them in terms of the blasts, whether the blast content in the bone marrow is less than 25% or higher in this ALL. But genetically, most of the mutations are very similar across the ones, except, of course, the ones that you're trying to understand. Is that correct in terms of that genetically the basal mutations between LBL and ALL are very similar, but they present very differently?
Yeah, that's correct. And we also compared our TLBL cohort to our TALL cohort, and it will be published soon in Blood Neoplasia, where we looked at the DNA differences and the activating pathways, and the general conclusion is that on DNA level they're quite similar. They're definitely quite similar. But Roland can maybe add something to that discussion.
Yeah, that is indeed true. If we look at the mutations or the mutational burden or something like that, they are very similar. This appears not to define the location of the disease.
I mean, so in your paper, you carried out whole transcriptome sequencing on 29 patients with TLBL, where you look for these, the genetic underlying or the mechanisms underlying the high-risk group. And you found a few new fusions actually in this paper, which is very exciting. And in this particular case, you used RNA sequencing to identify those fusions rather than, I guess, which a lot of people do is whole genome sequencing. What's the rationale for you to use RNA sequencing rather than whole genome, and what was your experience with that in this project?
Yeah, that's a good point. So we work in the Princess Máxima. There's a national center. We founded 8 years ago, and we then decided that we will sequence as much as possible with RNA sequencing all the tumors that we encounter. And that is also a bit of a luxurious position. We realize that. So we generate a lot of data to investigate those tumors. And so we did also for the TLBL. So when we had assembled quite a reasonable size of the cohort, we decided to look more in detail into it. And it's and that is how we discovered initially 2 cases that had this Notch1 fusion. And we were wondering what is going on. So how does this actually work? Does it lead to higher expression of the Notch compared to the Notch mutations, for example? And then when we looked more carefully to the expression of Notch throughout the whole cohort, we discovered that there were more fusions that were initially missed. And that is because they are actually promoter swaps, and they are not detected with the star fusion algorithms that we detected. So, and then it appeared that they are quite frequent, and that is basically how we discovered them. The whole genome sequencing data that have been included later, and then we see that there's also many similarities with but the differences are really in the fusions.
Yeah, that's personally I've had a long-standing interest in TLL, and I found it quite interesting that for TALL, activating Notch mutations, which occur in at least 60% of all cases and associated with generally good prognosis, and yet here in this paper you find Notch mutations in the high-risk TLBL. So, I mean, I guess, were you surprised by that finding? Because essentially, in both cases, the end result at the molecular level is that the intracellular section of Notch is cleaved by gamma secretase and then released. So the consequence is the same, and yet we have these very diverse outcomes for these patients. So, could this be, and I'm just going to prompt you, and from my thoughts, is it something to do with the cell of origin or the order in which the mutations are occurring? Because you could argue in TLL, the Notch mutations are quite late, whereas perhaps these fusions are truncal. And I'd love to get your insight into why in one case, the prognosis is good, and I guess the others is worse.
Yeah, that is the $1 million question, because initially we also were confused about this. So Notch fusion, is that the same as a Notch mutation, particularly because the Notch mutations are used into the stratification of those patients. But the difference, of course, is that Notch fusions lead to they lead to higher activity of the Notch, but still under control of the promoter of Notch1 itself. Whereas here in the fusions, a different promoter is driving it. And the initial analyses that we did also seem to appear that the Notch signaling as a whole is also much higher. So apparently, it's a matter of the level of notch fusion that makes some huge difference. So there is some increased level indeed leads to formation of a malignancy, which occurs in TLBL and TALL, with the extremely high level of notch signaling, and would occur a poor prognosis. But the details about that still have to be investigated.
Okay. So I can get that, yes, because that means that in terms of like TALL, we often get a range of mutations occurring. We have delta pest and HD mutations to drive that longevity of ICN, but the actual expression levels are not taken into account. Whereas I guess if you have a fusion partner driving very high expression, then that's another level of deregulation, which is played. And it's something I didn't actually consider in terms of the level of expression. So moving on, you've made a really nice discovery that the patients with these notch fusions have really high levels of the chemokine CCL17, which is also known as blood, thymus, and activation regulated chemokine. I had to look that up, or TARC. So tell me more about this finding and what made you look at CCL17.
Yeah, Jan discovered this. So
actually, that's quite a good question. It is because we have a long interest in CCL17 and not related to T lymphoblastic lymphoma. It is related to our Hodgkin patients. So in Groningen, another part of the Netherlands, they have a long history of investigating CCL17 or TARC as a marker for Hodgkin's disease. So we picked that up, I think, 5-6 years ago, and did several studies on Hodgkin lymphoma. So considering that our patients present with TLBL and Hodgkin lymphoma, sometimes quite similar, with mediastinal mass, but also with enlarged cervical or supraclavicular glands. So we standardly investigate all patients with mediastinal mass with CCL17 measurements. So actually, it was quite an interesting finding that when we analyzed our clinical data for our cohort of TLBL patients, that we found that they had these 7 patients mentioned in the paper, that they have hugely enlarged CCL17 values. So it was something we, to be honest, we accidentally found.
This is the best type of finding, the ones that take you by chance, but chance favors the prepared mind. So I guess the question is then, what's the source of the CCL17? Is it an autocrine loop? Is it a paracrine loop? Do you have any idea of its role in TLBL? Is it a consequence, or is it actually helping drive the disease?
We don't know much about it yet. We know from our Hodgkin studies that the Hodgkin cell produces the CCL17, so there it's clear. It attracts the T cells to the microenvironment. So the first thing we did was look whether the CCL17 was highly expressed in immunohistochemistry in our classes of our TLBL patients. But then it was not as impressive as we see it in Hodgkin lymphoma. So probably the T cells are not the ones who produce it. So until now, it's purely speculative what the source of the CCL17 is, and we know we're investigating that together with Roland to see whether we can find some clues where it's from. But to be honest, at this time point, we don't know yet.
The original data in staining we did not see a clue that the lymphoma cells produce it. But now that we have collected a little bit more cases and we look into the RNA expression levels, it does seem that the CCL17 transcripts are a little bit increased. But I agree with Jan, we have to look into this further because we don't know for sure.
Yeah, because I can imagine if it's actually helping drive the disease through a cytokine, chemokine receptor interaction, there is the possibility, of course, to exploit that therapeutically because there are CCL17 inhibitors, of course. So is that something you might want to, are you planning to pursue perhaps the introduction of these in a model system to see if the CCL17 inhibitors actually work at all, potentially? I don't know, can you make PDX models of these TLBLs and test that thesis?
Yeah, we're trying to initiate now the PDX models, and that's indeed what you need to investigate this. But for now, we consider it really as a biomarker, and also because of the potential of it. So I think it has great potential to discover those cases, and that is the most important now. If we can target it, it might be that CCL17 expression is somehow important for the strict localization of these TLBL. If you compare it to other TLL subtypes that also show bone marrow presentation, that might be. But if it's really associated with the poor prognosis, that's still a matter of further studies only to look into this.
Okay. If you look at the clinic, can I add something? Yeah. If you look at the clinical presentation of our TLBL patients, and I think it's for the ones who have the Notch1 fusion, it's quite interesting because they have massive mediastinal mass, but other groups have that as well within TLBL. A lot of pleural fluid, a lot of some had pericardial fluids, and most of them were very slow in getting into remission. So there is something resistant within this specific group. Not for all our relapses exactly the same thing, but it seems that the first phase, the first 2 months of treatment, that is insufficient for this specific Notch1 fusions. So it would be very interesting to explore other treatment options. So I think in future it's really interesting to look what the exact role of the CCL17 is, but also other thymic biomarkers. I would say.
Because I guess, thinking about Notch fusions, of course, the classic would be to use a gamma secretase inhibitor, which is not clinically possible because it's so poorly tolerated. But I guess if we could imagine a future where there was a Notch inhibitor or a gamma secretase inhibitor that wasn't toxic or better tolerated, is that something that may be useful, I guess? I don't know, Jan, what are your thoughts if that was ever possible or even feasible for these patients?
There is actually one who is well, one of the side effects of the MESH1 inhibitors which have been tested until now was that they have a lot of gastrointestinal toxicity, and it's based on a specific gene plays an intervention in this side effect. So there is one Notch1 inhibitor developed which does not actually has this specific side effect. So that is something we're exploring at the moment to see whether we can set up a trial or things like that to see first maybe preclinically, later on in patients. But that's a long story and still a lot of work to do. But it is possible, and I think there is actually one available.
Yeah, you mentioned the gamma secretase inhibitors, Charles, but that is the difficulty with that is, or at least there is no cleavage of those and not just intercellular domain fractions because of the fact that they only express cytosolically. So, and they directly produce. Most of these fusions use an alternative start methionine in the exon. It's very close to the gamma secretase cleaving site. So this Notch intercellular domain is also a little bit different. That might the gamma secretase inhibitors. I would not say that these might help. The Notch inhibitors might have potential. Also related to what I said in the beginning. Apparently, Notch activation can contribute to the malignancy, but it's really the excessive expression in the Notch fusions that drives this poor prognostic malignancy. That is what it looks like, and that also would mean that you don't have to completely inhibit Notch, but maybe lower it down already has a very beneficial effect. But obviously, this has to be studied in large cohorts. So we need to identify those patients and look into this.
Yep, absolutely.
And it will be a challenge, I think, to see what will be the combination. So you can, if you have a Notch1 inhibitor, which is usable in patients, then the next question will be how to implement them in a relapse kind of setting. That would be the first step, I would say. But maybe in future, if you speculate a little bit more and dream a little bit more, then maybe we can use it in upfront therapy for this specific subgroup as well.
Absolutely.
A lot of work to do, and what will be the backbone of the relapse protocol, and how to implement this Notch1 inhibitor is still, I think, things we need to discuss not only within our group, but also internationally.
Precision-guided therapy where we take
our LBL steering
the
committee.
genetics and then tailor the therapy of a child or a teenager with the therapy that's best suited for that child in terms of what the genetic makeup is, be it a fusion or another mutation. I think that's obviously the future, and perhaps even the start of what we could consider a chemotherapy-free future in a way, if we can only have, that's, if we really want to dream, I guess that's the ultimate future. So of course, in any paper, when you start your journey, it's never the same as how you wrote the paper. So the paper really reflects what actually happened in real life. And I was just wondering, in this particular case, if there were any unexpected findings or challenges or surprises that you had along the way, which then made you backtrack in terms of the findings and future work.
I think some of this topic we already discussed earlier in this podcast. The first moment we found the Notch1 fusion, the first one, we actually thought it was good risk. So our molecular biologists were convinced that these patients probably had the same mechanism as the Notch1 DNA mutations. So that was our first thought. And then, unfortunately, this patient relapsed and the second one relapsed. And then we started thinking, that's a coincidence. Now we started looking at all our patients, and from one thing led to another. So I think that was, for me, the biggest twist, from good risk to poor risk and to very poor risk in a few years.
Roland, any unexpected turns or aha moments for you?
Yeah, actually, our original question was, what is the difference between TLBL and TALL genetically? So that's how we started the study already years earlier, and collected a lot of sequencing data, and later incorporated also the RNA sequencing data. So that is also, there indeed we did not see many differences, except for those fusions. So it is indeed a larger project, and this is taken out as a key message because we see the prognostic and the clinical relevance of this, and it's so important. And therefore, we are so happy that this is had turned out into a single paper that really brings this forward.
It's a lovely paper. And so I guess, what's next? What's the short-term plan?
So to me, one of the first things we need to know is what is the percentage of relapses that actually hallmarks these notch fusions, and the other way around, how many notch fusions are actually present in a cohort of TLBL. So we need to systematically investigate the prevalence of notch fusions in TLBLs. And for that, we are now investigating heavily in different techniques to identify those fusions effectively. That is targeted sequencing approaches, but also break apart FISH. And of course, I think the TARC screenings, the biomarker, can help us quite a bit with identifying those fusions.
Great. Well, I think we can wrap it up there. Thank you both for such an insightful discussion on your paper, and congratulations on your manuscript, which is now published in the Hemisphere.
Thank you very much.
Thank you very much for the opportunity.
Thank you for listening to this episode of the Hemisphere podcast. All of Hemisphere's content is open access and can be found at www.hemispherejournal.com. We hope you will join us for future podcast episodes.